Good evening and welcome. I'm Mike Botchin, the Dean of Biological Sciences and professor in the Department of Molecular and Cell Biology here at Berkeley. I'm really delighted to be here to talk about a topic that may not be top of mind, but that is relevant for all of us. That's drug resistance. This topic has been overshadowed recently by current events, but our moderator and speakers will make it clear why this is an urgent and troubling problem from a public health perspective, as grave as viral pandemics. Through the wonders of modern medicine, one may receive wonderful treatments in our hospitals for complex medical conditions, but fall fatally sick from infection with the drug-resistant bacteria in such hospitals. We shall barely scratch the surface tonight, but our aim is to show you some of the great work and great researchers here at UC Berkeley who are devoted to drug resistance and to solutions. So this evening, we're going to hear from 4 scientists who are going to frame the problem for you and offer a number of perspectives based on their research and expertise. Our moderator this evening is Michael Marletta, a professor of chemistry, a professor of molecular and cell biology, and the C.H. and Annie Lee Chair in the molecular biology of diseases. Professor Marletta is a distinguished member of the National Academy of Sciences, the American Academy of Arts and Sciences, and the American Philosophical Society. Michael leads a group at Berkeley focused on understanding, from a chemistry and molecular biologist lens, the study of protein function and enzyme mechanisms, seeking molecular answers to complex problems in biology. Michael, take it away.
Thank you, Mike. It is a pleasure to be here this afternoon, and I also want to thank all of you for attending. Give me one moment here to get a little bit organized. Let me get my laser pointer. Okay, so Mike what am I going to do? What I'm going to do in the first few minutes is attempt to sort of place some boundaries on the kind of discussion that we're going to have with my 3 colleagues that follow. So I just want to sort of outline the problem and give you an idea of what to expect over the next few minutes. So Mike alluded to the fact that we're, maybe by current events, he meant we're kind of overwhelmed, as well we should be, with COVID-19 and the pandemic associated with it. And here's some numbers from the CDC from last week, and you can see the numbers are indeed large, including the one that of course is most concerning, of the number of deaths in the United States. But that's actually not the pandemic that I want to talk about today, and that I'm going to provide the boundaries for the rest of the discussion. But instead, it's, as Mike Botchin just said, bacterial infection. So here's a picture of Staph aureus, a very common pathogen in humans. This particular one gets its name, methicillin-resistant Staph aureus, MSRA, because it is indeed resistant to this very commonly used antibiotic methicillin. And you can look at the numbers, granted they're from 2013, and you can say like 11,000 deaths. These kinds of numbers are the ones that attract more of my attention. Why should we be worried about this? And we should be worried about it because, as the name of the organism that we've given it, it's resistant. And in that context, we'll come back to these, what you might think right now are small numbers. So that the public's aware of this. I'm not going to be surprised if we let you all speak that some of you would say, yes, of course I've heard about this. And here's a variety of media sources that have talked about this problem and continue to talk about this problem. But let me sort of try to put it into a more definitive context. Here are the list, and they're by brand name antibiotics, the top 10 that are prescribed in the United States. And I'm sure you can look at this and see 1 or 2 or 3 or more that you have taken over your lifetime. Every one of these has high-level resistance in the human population. And that brings me back to that small number that you saw on the first slide. And let's now look at estimates. So these are worldwide now, deaths per year estimate from antimicrobial resistance. So to make it more clear, in this part of the wheel, deaths from pathogens that are resistant to antibiotics. And the estimate for that in 2050, which is not that far away, is 10 million people on the planet every year. Compare that to the number of deaths from cancer. It is already now significantly larger than that. And you can look at a variety of other diseases or road accidents, for example, to see that that value far outstrips any of those other numbers. So while small now, it's a problem that's going to continue to grow and grow every year, ultimately getting to numbers, projections of 10 million people in 2050. So that's the microbial side of things. What about the antibiotics themselves? Well, that's the other part of the problem. So what you're looking at here is the number of antibiotic classes discovered or patented, starting in the 1890s and working our way up to at least the 2010s. And it's pretty obvious that there was a golden age of discovery in the 40s, 50s, through even into the 1980s, but not much since. And you'll hear why in the talks this afternoon. Some of that will become clear as to why that dramatic fall off has occurred. But this really now brings me to the end of what I wanted to say, which is the problem. The problem is twofold. It's resistance to the antibiotics themselves, but the additional part of the problem, just as significant, is that we don't have any new ones to combat those resistant organisms. So that's the kind of scope of this problem that is continuing to grow across the planet, and what you'll hear about this afternoon. So, in my opinion, Berkeley's greatest strength is its commitment to discovery and its ability to solve big problems. And I've outlined a big problem. Indeed, it is a very big problem. So you're about to hear from three of my colleagues with approaches to this problem that exemplify the very best of Berkeley in both discovery and tackling and solving big problems. So with that, I will turn to my colleague, Art Reingold. Let me get the screen ready for him. So Art is a professor in the School of Public Health, and he's the division head of epidemiology. His research interests focus on vaccine preventable diseases in the US, particularly in low-income settings. He also focuses on emerging and re-emerging infections, not only in the United States, but globally, and prevention and control of respiratory infections, including influenza and COVID-19. Art, stage is yours.
Thank you. Thank you very much. Michael, can you see my slides?
I
Yes.
can.
Excellent. So I'm going to give just a little bit of history and then talk a little bit of our work in studying antimicrobial resistance, but a little bit of history first, or definition of a couple of terms. So it's of interest to some people perhaps that the word antibiotic is quite an old word, and it really is meant to refer to products, natural products made by fungi or molds that can suppress or the growth or kill other microorganisms, as opposed to antimicrobial agents, which include antibiotics, but it can be synthesized in the lab and are not necessarily natural products. And just to go back to what Michael was talking about, we define antimicrobial resistance as when microorganisms change in ways that render the medication used to cure the infections they cause ineffective. And that's the clinical problem that Dean Botchin was referring to, is you have an infection, we need to treat it, and we don't have any antibiotics or antimicrobial agents that are effective against your infection. So just to point out in this one very quick slide that people have known since ancient Greece and Egypt and India that molds and fungi and plants contain substances that will help heal wounds and keep them from getting infected, and have used molds and plants and mixtures of wet bread and spider webs and the like to treat wounds. But it really wasn't until the 1870s to the 1920s that scientists like Lister and Pasteur showed in the laboratory how molds inhibited bacterial growth. Of course, in a more modern context, in the last 100 years, will almost be the 100th anniversary of when Alexander Fleming concentrated the first natural antibiotic, penicillin, from a mold, and there are cute stories about having left a petri dish near an open window and all of this being inadvertent. And around the same time, scientists in Germany were working on the first antimicrobial agent, not from a mold, the sulfonamides, or a drug called Prontosil. I would point out that Fleming, together with Florey and Chain, won the Nobel Prize for discovery and developing penicillin in 1945. And I now want to point to a rather prescient statement that Fleming made in his Nobel lecture when he said that the time may come when penicillin can be bought by anyone in the shops. Then there is the danger that the ignorant man may easily underdose himself and by exposing his microbes to non-lethal quantities of the drug, make them resistant. And some of you may know that there are parts of the world where you can in fact buy antibiotics over the counter in a drug shop. But the point here is that he foresaw the time when drugs like penicillin might not work anymore and that the microorganisms might evolve to resistance. Michael showed you the distribution of discovery of new antibiotics and the fact that it peaked in the 1950s. This is a cartoon after the Salk vaccine report was issued, where medical history is crossing various diseases off his list. And while 3 of these, smallpox, diphtheria, and polio, were due to the development of vaccines, pneumonia being crossed off here is meant to refer to the fact that we had penicillin and other antibiotics to treat pneumonia, not to the development of any vaccines. So by the 1950s, there was great enthusiasm that infectious diseases were largely a thing of the past. Oops, sorry about that. Now, he gave you a list of various brand names of antibiotics and antibacterial agents. This slide is simply to show you the broad categories of antibiotics that we have for treating different bacterial infections. And I'm not going to get into the details about these drugs, although we will be talking a little bit later. Richmond will be talking about the beta-lactamase or the beta-lactam ring in certain bacteria. Again, antimicrobial resistance is quite complex. And just one broad category of bacteria, the gram-negative bacteria, this is just one set of the molecular approaches that microbes have to either blocking the actions of antibiotics, inactivating them, or keeping them out of the bacterial cell. And I would just point to the beta-lactams here in the middle, because Richmond will be talking about those more later. And again, not to steal his thunder, because he understands beta-lactam rings and I don't, but again, this is simply to show that the microorganisms have multiple ways of developing resistance to the various antibiotics. In this case, the beta-lactamases, which can inactivate a drug like penicillin. So you also heard from Michael that the world has been growing concern about this problem. The CDC, the NIH, and the World Health Organization have all issued reports about this threat of antibiotic or antimicrobial resistance over the time period, in this case from 2013 to 2014. And in these reports, this is a more recent WHO report about which bacteria we need to be most concerned about and which are the critical. And I'm going to talk a little bit about the Enterobacteriaceae that are shown here and their development of resistance to a 3rd generation cephalosporin. But there are certainly other bacteria that we are quite worried about with regard to resistance. And so here you can basically see that these organizations talk about urgent threat pathogens, but just to move on a little bit, They also talk about urgent threats. So in this particular report, some of you may have heard of Clostridium difficile, an infection that you get often in the context of getting antibiotics for a different infection, which then lead you to develop this very serious kind of diarrhea, which can even be fatal sometimes. I'll talk a little bit about the carbapenem-resistant Enterobacteriaceae. And on the right-hand side here is drug-resistant gonorrhea. And fundamentally, the world really only has one antibiotic left for treating drug-resistant gonorrhea. So we're in pretty dire shape when it comes to that disease. There are other bacteria. Sarah Stanley will talk about TB and drug resistance in TB. Michael's already referred to the methicillin-resistant Staph aureus, which has been in the past a huge problem in hospital and healthcare settings with transmission in those settings as well as in the community. But again, there are many different bacteria that we are worried about. There are a number of strategies for preventing and controlling antimicrobial drug resistance. Some of these, the first three really refer to what clinicians do. The second and third refer to what clinicians do with regard to how antibiotics are used and bundled together with other interventions. Some of what we need to achieve can be done by reducing misuse of antibiotics in animals, which is a very challenging area. I'm going to focus my comments about surveillance and vaccination and their role in studying and combating antimicrobial drug resistance. But I just want to point out that antimicrobial stewardship here basically refers to the fact that if we do get a new antibiotic that's effective against some of these resistant bugs, we want to keep it locked in the medicine cabinet and use it as sparingly as possible in order to reduce the chances of inducing resistance against some wonderful new antibiotic. And the problem with that, while that's what's important to do from a clinical point of view, it's not so good for making a profit on what a drug that might have cost $1 billion to develop and bring to market. So just to get to my own work, I run a program funded by the Centers for Disease Control and Prevention. I've been doing it for over 25 years at this point, the California Emerging Infections Program. And we study quite a wide array of different infectious diseases here in the Bay Area. The current budget is over $7 million, although about half of that is new funding to study COVID. But one of the things we do is we conduct high quality surveillance for various infectious diseases and studies of those diseases. And included among those are the antimicrobial resistant infections of most concern. So just to show you an example here, Michael referred to the methicillin resistant Staph aureus, MRSA, and those are the orange bars here. And you can see that we track this very carefully. And if you look down at the bottom, what you'll see is that infection with the resistant organisms is associated with a number of worse clinical outcomes. And that's what we're really concerned about. I would simply point out that the reason there are no data here for 2019 and 2020 is that the staff who normally would be completing the collection of all these data are so busy with COVID that we haven't been able to finish that work for the last year or so. And so here's an example of these carbapenicillin resistant Enterobacteriaceae that we monitor in the 3 Bay areas. And these are the various bacteria that are resistant to pretty much all of the drugs we normally would use to treat these types of infections. So they are not uncommon. These are from hospitals here in the Bay Area of hospitalized patients, and they pose a real challenge in terms of treatment and preventing their spread. Finally, I just want to quickly point to the fact that vaccines can be helpful to us in dealing with antibiotic resistance. So this is a graph showing data from our site and other sites that we work with after the introduction of a new vaccine in children to prevent what we call pneumococcal infections, a very common cause of pneumonia in kids, and showing that when the vaccine was introduced in the year 2000, the number of children vaccinated went up quite quickly because pediatricians are very good at vaccinating children for the most part. This resulted in a substantial decline, as shown here on this graph, in the number of these infections in children in our surveillance areas, but also, in fact, in adults, because children are a major source of infection through person-to-person transmission of these organisms to adults. So we had a massive decline in these infections, but with regard to antibiotic resistance, we saw a huge decline in infections caused by antibiotic-resistant Streptococcus pneumoniae, the pneumococcus. And that's basically because the types that are in this vaccine are the ones that are most likely to develop antibiotic resistance. So vaccines can be one tool that we use as we're trying to deal with this problem. So in summary, antimicrobial drugs save lives. We really need them. Antimicrobial drug resistance is a major global threat, and we desperately need new antimicrobial drugs to deal with some of these emerging threats. Thank you very much.
Thank you, Art. We have a couple of questions that were posted. So the first is from Ken Hart, and he asks, is there a possibility of using CRISPR to target bacteria to chop up their DNA and/or RNA?
I'm going to defer that question to my colleague, Sarah Stanley, who knows more about CRISPR than I do. I'm privileged to be in the building next door to the kingdom of CRISPR, but I'm not the right person to answer that question.
As it happens, go ahead, Sarah.
I was just going to say, I think that's a great question. And in theory, yes, it could be possible to do something like that. The major challenge for using CRISPR as kind of an antibiotic is delivery and getting it into a bacterial cell where it needs to be during an infection.
I was going to add that as it happens, I'm teaching a graduate course in the afternoons, and from 2 to 3:30 this afternoon, our guest lecturer was Jennifer Dowden, and Jennifer actually addressed that issue specifically and mentioned including some of the challenges that Sarah just mentioned. Okay, Brian Majorella asks, is the gap in development of new antibiotic classes the result of our switch to the application of advanced molecular discovery techniques?
I don't work for the pharmaceutical industry, so what inhibits their research on additional antimicrobial agents, I don't know, but there's no question that part of it does have to do with whether they can recoup their development and research costs of bringing a new drug to licensure in the United States, which can easily be $1 billion or more. And if people like me are then going to keep the drug in the medicine cabinet and allow it to be used as sparingly as possible, that sort of goes in the opposite direction to how they can get back their development costs. So, but I don't honestly know what their other impediments are to either discovery of antibiotics in the natural environment or to the creation of molecules in the laboratory, which Richmond is probably better suited to talk about.
Okay, thank you, Art. Let's move on, and we'll move on to Sarah Stanley. We just heard from her briefly a moment ago. So Sarah is an associate professor in molecular and cell biology and in the School of Public Health. And her research interest mainly centers on the study of tuberculosis, where she's studying mechanisms of immune control and pathogen evasion of immunity. She uses a wide range of techniques and approaches. Her long-term goal is to better understand host pathogen interactions, allowing for the development of novel therapeutics. Most recently, she's also made a pivot to research connected with the COVID-19 pandemic. Sarah.
Thank you, Mike, for that introduction. And also thanks to Art for that really wonderful introduction to kind of the backdrop of the problem of antimicrobial resistance. So today I'm going to focus on one infectious disease in which drug resistance presents a major threat to public health around the world, and that is drug-resistant tuberculosis. So we've obviously all been living through the global COVID-19 pandemic, and this pandemic has caused an estimated 3 million deaths worldwide to date. And for many of us, we really think of this as kind of our first experience of a pandemic, which of course is defined as an epidemic of infectious disease that is spread across a large region of the world. However, we actually live in the midst of several major but slower moving pandemics, including HIV, which I think we're all familiar with, which emerged in the 1980s, and also tuberculosis. So tuberculosis, unlike HIV, I think there's less awareness of the importance of the TB pandemic for global public health. And many of us in the United States are really familiar with tuberculosis only as a disease of history. In the 18th and 19th centuries, when TB was called consumption, it was responsible for 1 out of 4 of all deaths. We've made a lot of progress since then. However, tuberculosis today is still the top cause of death by infectious disease every year globally, with the exception of last year during the COVID-19 pandemic. And it's estimated that 2 billion people are infected with TB worldwide, and that 1.4 million people die from this infection every year. So most of these infections are concentrated in low and middle income countries, with in particular sub-Saharan Africa having the highest rates of infection, which is why many of us in the United States are not even really aware that TB is still a major problem today. So tuberculosis is a disease of the lungs, and as with all infections, the immune system, which I'm representing here with these purple cells, come to the lungs to try to fight the infection and kill the bacteria. Now, ideally, the immune response is able to control the infection and preserve the health of the host. And I've kind of depicted that here as a productive immune response leads to elimination of the bacteria and then ultimately resolution of the immune response. However, in the case of what we call active tuberculosis, the bacteria become really impervious to the immune response. They continue to grow in the lungs. And as the number of bacteria increase in the lungs, you get an increased immune response that can ultimately become so powerful that it starts destroying the lung tissue in a failed effort to kill the bacteria. So this is actually analogous to severe disease with COVID-19, in which it's actually an inappropriately strong immune response that results in pathology and death of the patient. So for TB, thankfully, we have antibiotics that can kill the bacteria. This eliminates the stimulus that results in an overactive immune response and restores the lung to a normal, healthy state. So antibiotics are really crucial, in the fight against the TB global pandemic. Unfortunately, as Art alluded to in his talk, the improper use of antibiotics has led to the emergence of drug-resistant TB. And one of the problems that is somewhat unique to TB is that it's incredibly difficult to treat, even in the best case scenario where the bacteria are not resistant to the antibiotics. So standard TB treatment requires 4 antibiotics for a minimum of 6 months. You have to take a handful of drugs every day. These drugs have significant toxicity. They can make people feel really sick and can even cause liver damage. So what happens in parts of the world where you can just go buy TB drugs over the counter, or potentially also in the United States, if a doctor isn't closely monitoring the patient will start to feel better after taking the drugs for a short period of time. They'll stop taking their drugs, and then that provides the opportunity for drug-resistant bacteria to emerge. So every year, about 500,000 people are diagnosed with drug-resistant TB. If you have drug-resistant TB, this requires lengthy, years-long drug regimens with even more toxic drugs, and half of patients with drug-resistant TB ultimately succumb to their disease. Something that's really scary that has emerged in the last few years is totally drug-resistant strains of TB that are refractory to all existing treatment regimens. And this raises the specter of TB spreading that is completely unable to be treated by modern antibiotics. So in order to understand the problem of resistance, let's take a moment to talk about how antibiotics work. So bacterial cells, like all cells, need to carry out the fundamental processes of life, which means they need to take in nutrients to grow, build the components of the cell, and ultimately divide and reproduce. So these essential processes are carried out by proteins, which I've depicted here in these kind of blue and green shapes. And so proteins will carry out kind of these essential processes in the cell. And what an antibiotic will do is it will enter the bacterial cell and it will physically attach to one of these essential proteins. And what it will do is stop the protein from carrying out that function, which means the bacterial cell will die. Unfortunately, bacteria have numerous strategies for becoming resistant to antibiotics. Bacteria are really smart and good at evading traditional antibiotics. So first, they can mutate their DNA so that to change the shape of an essential protein so that that protein actually can still carry out its essential function for the cell, but the antibiotic can no longer bind to it and inhibit its function. That's called binding site mutation, and it's the most common strategy used by TB. However, there are many, many other things that bacteria can do, which Art alluded to. They can acquire genes that encode proteins that allow them to cut the antibiotic so that it no longer functions, or chemically modify it so that it can no longer bind its target. It can acquire genes or upregulate the expression of these little proteins that act as kind of molecular pumps that actually pump the antibiotic out of the cell. Once a bacterium becomes resistant to a specific antibiotic, there's really nothing you can do except for find another antibiotic to treat that resistant infection with. So this is why what Art discussed is that we're in a kind of a constant race against antimicrobial resistance, where as soon as a new antibiotic is widely used, bacteria will develop resistance to it. And so you need a constant entry of drugs into the pipeline for antimicrobial discovery to stay on top of the problem of drug resistance. So one question that my research program addresses is whether we can find ways to circumvent this kind of arms race of antimicrobial resistance by coming up with totally new strategies for treating antibiotic-resistant infections that really circumvent the normal mechanisms by which bacteria become resistant to traditional antibiotics. So I'm going to tell you a little bit about one project in my lab in which we started by thinking about, if you remember, I told you that one of the problems with tuberculosis is that in the case of active disease, the immune system is unable to control the infection, and that part of what kills the patient is an attempt to maximize the immune response to kill the bacteria. The immune system also increases an inflammatory response that destroys the host tissue. So we're really interested in the question of what if we could design a therapeutic that's able to boost the beneficial aspects of the immune system, killing the bacteria, without causing the collateral damage to the lung tissue that can result in morbidity and mortality from the disease. So this kind of therapeutic is called a host-targeted therapeutic, because instead of targeting the bacterium for killing, it targets the host, so the patient, and theoretically avoiding the problem of antibiotic resistance. So how can we go about designing such a therapeutic? So the cell type that my lab focuses on is an immune cell called a macrophage, which I've depicted here in gray, because the macrophage is crucial both for making proteins, depicted here in blue circles, that actually go to the bacteria, bind to them and kill them, or make products that can directly kill the bacteria, but also for making the inflammatory proteins, which is the part of the immune response that can be detrimental if it's too strong. So the problem with trying to influence the immune response to enhance this part that we want, the bacterial killing, is that these responses are usually coupled. So whatever you do to enhance bacterial killing will also enhance inflammation to the point where it could be detrimental to the host. So you might ask, well, we have tools like steroids, which suppress inflammation. Many of you are probably aware that that's one of the kind of blunt tools that people try to use for treating severe COVID-19. But the problem is that when you use a steroid to depress this inflammatory response, you run the risk of weakening it too much, and you do need some inflammation in order to combat the infection. So what you need is to maximize bacterial killing while maintaining just the right level of inflammation. So how do we do that? So my lab has discovered that a protein called hypoxia-inducible factor 1 alpha, which is abbreviated as HIF1 alpha, is a very interesting regulator of macrophage responses to infection. Now, it's possible that for some of you in the audience, HIF1 alpha rings a bell because the scientists who discovered HIF1 alpha in the context of its normal function, which is cellular responses to low oxygen environments, won the Nobel Prize in Medicine for this discovery in 2019. So surprisingly, we've discovered a role for HIF1 alpha in infection. And what we have found is that HIF1 alpha has just the qualities we would ideally want in a host-targeted therapeutic for TB. HIF1 alpha can actually activate specifically the expression of these proteins that have the beneficial activity of killing the bacteria, while not aberrantly also activating at the same time the overproduction of these inflammatory proteins. And so what I'm showing here is that if we take cells that are infected with TB, normally the cells would, this is bacterial numbers in these cells here in the blue line. Normally the bacterial numbers would either go up or stay steady. And if we activate HIF-1 alpha with a drug, so interestingly, there are lots of drugs that are kind of under development for HIF-1 alpha activators outside of the context of the infectious disease that we're able to sort of repurpose for TB. If we add one of these drugs, we can decrease the bacterial number in these cells and induce the host cell to kill the bacteria while having very minimal effect on the inflammatory response. So we think that HIF-1 alpha activators have really interesting potential for a host-targeted therapeutic for TB and possibly other bacterial diseases. And what we're doing now is testing these inhibitors in animal models as a proof of concept that we would need before considering moving into human patients. But in general, this is just one strategy that you could think about taking for targeting the host and potentially allowing us to develop therapeutics that circumvent this problem of resistance to traditional antibiotics. Okay, so with that, I'd like to thank my lab, and in particular, a very talented graduate student, Jonathan Braverman, who started this work in my lab, and thank you all for listening.
Thank you, Sarah. We also have a couple of questions for you. The first from James Felton, who would like to know, what can you tell us about research on using bacterial viruses to control bacterial infections?
Yeah, that is a great question. Phage therapy, so phage are the viruses that, that can actually directly, specifically identify bacteria and kill them. So phage therapy hasn't, I think, in a lot of people's opinion, gotten enough kind of attention, but that's changing. And actually, there was just a really exciting report of a patient infected with another mycobacterial infection, Mycobacterium abscessus. Now, abscessus is really rare, but it's very, very scary because it's just super, super, super drug resistant. And these researchers identified a phage that killed abscessus. They treated the patient and it was successful. So I think that we're going to be hearing a lot more about phage in the future.
Okay. From Natalie Shuttleworth, she would like to know what is the role of our immune system versus antibiotics to combat microbes? Can we strengthen our immune system or change our microbiome through diet, for example, in lieu of taking antibiotics?
Yeah, so the immune system is really crucial. So even when you take antibiotics, really what you're seeing is a collaboration between the antibiotics, which can really knock the bacterial numbers down, and then the immune response, which can kind of eliminate, help to lead to complete sterilization of the infection. Maybe I should defer the question about how we can strengthen our immune system through diet to Art, who is, an MD. But what I would say, as from the basic science perspective, is that there are no clear kind of paths in terms of the microbiota to really manipulating or boosting the immune response. Although, of course, like a healthy diet, all of that is definitely beneficial for the immune response.
We are getting close to the time where we should move on, but there are three more questions. Sarah, I don't tell you that, so maybe you can give short answers now, and so that we can at least have everyone get their question in. So Randy Shingai says there's been a lot of talk about vaccine-resistant variants of the coronavirus. Why hasn't that occurred, or has it, for bacterial vaccines?
Yeah, can I answer this in the chat, actually? Because I have a kind of a longer response to that then.
But maybe Warren Gish's question is a shorter answer. Could you use steroids plus HIF-1 alpha induction?
Well, our hope is that we wouldn't need to use steroids with HIF-1 alpha induction, because HIF-1 alpha has this kind of natural property of putting a brake on the inflammatory response and keeping it from being too excessive.
We have, I fear, a philosophical question with a long answer from Brian Majorella, where he says, why did evolution, under centuries of TB pressure, not decouple T-cell bacterial killing and inflammatory signaling?
Yeah, I mean, that's a fascinating question. And I guess what I would say to that is that, evolution is an ongoing process, and what we're seeing is a snapshot. And what I talked about today is the 10% of people who get active disease who are infected with TB. Didn't talk about is the 90% of people who have exactly an immune response that can suppress the infection, probably not eliminate it, but suppress the infection without causing excessive inflammation.
Okay, and you have one question I'm gonna hold you to answering in the chat. Thank you very much, Sarah. So it's my pleasure next to introduce our third participant of my three colleagues today, Richmond Sarpong. Richmond is a professor of chemistry. He's also an executive associate dean in the College of Chemistry. Richmond's a synthetic organic chemist, and he and his students synthesize beautiful and complex molecules found in nature that are termed natural products. Through the synthesis of natural products, Richmond finds inspiration for new medicines and develops methods and strategies to synthesize new therapeutics. Richmond, the screen is yours.
Thank you very much, Michael, for the warm introduction. It's a real pleasure to be with all of you this evening and to have an opportunity to tell you a little bit about what my laboratory does. And to do that, I thought I would start at the beginning. And so here's a picture of me at 5 years old, and I was born in Ghana in West Africa, and we've already seen a map of the world. And so that gives you a little bit of context. And growing up in that era in Ghana, I actually contracted malaria 3 times as a child. Now that's a parasitic disease, but that's really what launched me on my journey to become a chemist. And a lot of that was also due to another parasitic disease called elephantiasis, which is caused by this adult black fly, which bites and leaves behind a parasite in the human body. And unfortunately, that parasite travels to the eye membranes and it leads to blindness. And so this is also known as river blindness. Now, that is not the reason why I got interested in chemistry. The reason I got interested was because of the worldwide response to this particular disease, which was that the pharmaceutical company, Merck Research Laboratories, and an institute in Japan called the Kitasato Institute developed this particular compound called ivermectin to address this parasitic disease. Now, this is an example of a natural product. And this particular natural product, which comes from an actinomycete bacteria, was isolated from a very unusual place, which is that it was isolated from a golf course. And over the billions of years, nature has had an opportunity to evolve molecules like this to address disease. And so this is the exact golf course from which this molecule was isolated in Japan. It was isolated by this scientist called Satoshi Omora, who ultimately won the Nobel Prize in Physiology or Medicine in 2015. And I've had the opportunity to collaborate with him and his team in more recent times. But this is what really got me interested in these natural products. Now, if you've not heard of ivermectin, you're forgiven. But one molecule that you've certainly heard about, which is an inspiration for why natural products are the basis for a lot of medicines, and that's illustrated here in this particular statistical model, is penicillin. And so natural products like ivermectin and penicillin have been the basis for medicines for a very long time. Now, Professor Reingold has told us a little bit about penicillin here today. It's isolated from a fungus. And in fact, the Petri dish that he mentioned here is an exact picture of that Petri dish as was published by Alexander Fleming many years ago when this was discovered in 1928. And so Fleming, of course, went on to win the Nobel Prize, as was described earlier. And penicillin was first used to address a huge infection, which was caused by a deep cut to the face of this particular person, Albert Alexander, who was a police officer. Now, it turns out that penicillin was effective in addressing the disease, even though they had used impure amounts of this penicillin drug. And so after 5 days, they exhausted their supply. And unfortunately, the patient died. However, this result of success, because prior to that time, that type of infection was a death sentence. So this result really sparked collaborations across the world to really develop penicillin into the drug that it is today. But as you've also heard today, it turns out that unfortunately, because of the misuse of a lot of antibiotics, they develop resistance. And in addition to that, the development of antibiotics, because that's not a lucrative undertaking, that has been challenging to develop new antibiotics. But despite challenges, there have in fact been new antibiotics that have been developed that are based on natural products like ivermectin and penicillin that I told you about before. And so on this slide, I show a few examples of these types of antibiotics, which are useful. However, as we've noted here today, there's resistance that's been growing to these antibiotics. But my laboratory has been especially excited and inspired by penicillin and its related derivatives. And so, for example, here is a derivative of penicillin. And one of the things that I didn't talk about is the structure of these molecules. And so here I've highlighted this four-membered ring containing a nitrogen, which, as Professor Reingold mentioned, is a beta-lactam. And that is an important warhead to this molecule, which reacts with the bacterial cell wall and ultimately leads to the death of the bacteria. And so it turns out that this derivative of penicillin was developed by a pharmaceutical company called Eli Lilly in 1964, but it was shown to be effective only against thin-walled bacteria. Okay, those are easier to kill. And over the course of time, there have been other derivatives that have been made, such as this one here. You can see that it bears the same warhead. However, there have been changes to the periphery of this molecule, and that in fact imbues this molecule to be active not only against these thin-walled bacteria, but also thick-walled bacteria, which are much harder to kill. And so what that has taught me is that if we're able to make very deliberate and intentional changes to the periphery of these molecules, we can in fact imbue them with new activity. And so that is something that my laboratory has been working toward. And we've been interested in developing first a very precise way of putting together the core of these molecules. And in our development, we were very interested in utilizing chemical synthesis to then make the changes to the periphery of these molecules. Now, when one thinks about chemical synthesis, we're often sort of met with visions of, belching stacks of smoke coming out of smokestacks and so forth. But in the modern era, we've been interested in taking advantage of sustainable ways to make molecules, okay? And so we have been especially drawn to utilizing sunlight as a reagent to make molecules. Now, sunlight is free as far as I can tell, and is for the most part innocuous. And so the development that we have undertaken in my laboratory is to take precursor compounds of this type, which are easy to access, and simply by treating them with sunlight, we're able to effect a change where we effect a connection between these two, and that in fact builds that four-membered beta-lactam ring. And so in that way, we can easily prepare the core structure, which is very much related to the types of molecules that I've been telling you about, which are inspired by penicillin. And so we are very hard at work making changes to the periphery of this core structure, which will hopefully imbue that particular core structure with the types of activity that one would like to see in a new antibiotic. And our laboratory has had a lot of experience in terms of making these atomistic changes to molecules of this type over the years. And so this is work that we're very excited about in the context of developing potentially new antibiotics. And so I will stop there and thank my research group, which I'm very proud of. It's a very diverse group of individuals that are undertaking this work and all the foundational work that has led to the little snapshot that I was able to give you here today. So I'll pass the mic, if you will, back to Mike.
Thanks, Richmond. So there is a question from Rick Reeder, and he asks, given the forecast of many TB deaths, wouldn't a treatment be lucrative enough to pay for its development?
Well, so I guess in terms of TB, I'll certainly cede the floor to Sarah Stanley. But what I can tell you in general about the sort of landscape, as far as I understand it, with developing new antibiotics, is that, in fact, when you have a very, 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 very active antibiotic, which is sort of the last bastion of defense, in many ways, the federal government will probably pay you to hang on to that, to defend against, incurable disease. And so there perhaps might be models there for developing these antibiotics, but we need a lot of them and a wide range of them. I know Art is an expert on this, and so I'll also let him speak about this.
So if I could just give a brief answer from a different point of view, as Sarah pointed out, TB is a disease of the poor. And the overwhelming proportion of people with TB are poor people. And they can't afford to pay very much for drugs. You may know that in the COVID circumstance, we're trying to raise, or something called COVAX is trying to raise $40 billion to buy COVID vaccines and treatments and the like for people in low and middle income countries who otherwise would have no access to these because they can't afford them and their governments can't afford them. So it's just a problem of poverty, basically.
Richmond, maybe I could ask a question. The vast majority of all of the antibiotics, and you highlighted nicely how many of them are natural products, but the vast majority of natural products come from a handful of organisms that we have cultured in the lab and studied and learned how to extract the natural products from. What's the strategy for looking at this wide expanse of other organisms on the globe?
Right. So, there are, and this work is happening at Berkeley. And so Michael, well placed in terms of asking this question. But, there are ways to think about perhaps mining the genome of other organisms to identify those organisms that might produce a particular natural product, which could be effective. Because as you may know, a lot of these molecules that I talked about are produced because of evolutionary pressures. And so often it's under stressful situations that these genetic machineries are sort of turned on to produce a lot of these other natural products. And so it might very well be that if we actually mine the genome of organisms, there might be silent genes that represent much more active compounds. And there's a lot of exciting work that's happening here at Berkeley in that regard.
Okay. Mark Benzadoun asks, how do you decide what structures to add to the central nitrogen ring?
That's a great question. In fact, what we look for is methodology where we can really vary widely the types of structures that we add to that central ring. However, one can also look at co-crystal structures of how these molecules bind to their host, biological host. And on the basis of the types of contacts that you need to be effective, you can start to rationally design the types of atoms that you add to establish different connections to better recognize, for example, the biological host. So we take a two-pronged approach, one of which is rational, based on existing co-crystal structures, and the other is, I would say, not irrational, but very diversifying, where we look to address a wide range of because there are often times when you have a combination of these atoms and they take on a life of their own. You can say emergent properties, and so it's sometimes not easy to predict which particular combination will lead to the most effective compound.
Thanks, Richmond. We are getting close to the ending time here. There was a question posed by Tom Klein that is under Richmond, but I think it could be one addressed to any of us. And it says, with mRNA vaccines being now so quick and easy to make, has it become more economical to consider vaccine approaches to killing most medically dangerous drug-resistant bacteria rather than drugs per se?
think I'll see the floor to Art or Sarah.
Maybe Art can handle that.
So it's a great question. And there's been a lot of work in recent years for vaccines against various bacterial infections. Staph aureus would be one good example, but these gram negatives are another. It's not quite so easy to make a vaccine that offers protection against these bacteria. So far, the main problem is people have not come up with safe and effective vaccines, but there's certainly a lot of work going on in that area.
And the last, I think we do have time to sneak in one general question to all of us. From Ken Harden, it again addresses the delivery issue with CRISPR. And Ken asks, can nanolipids be used to breach the bacterial cell wall to deliver the appropriate CRISPR package? I think what I will say, and there's silence here, Ken, you hit upon, even as I mentioned, Jennifer talked to my graduate class for an hour and a half this afternoon, and delivery was a significant piece of what she spoke about. Getting these proteins to the right place, how to encase them, how to deliver them with specificity is indeed a major challenge, and one that remains as, for the key deployment, broad deployment of CRISPR as a therapeutic. That certainly is the issue. I need to bring this to a close because we're going to run out of time. My rather longish introduction from my colleague and friend Mike Botchin will now be shortened quite a bit so he can provide some closing words. Before I introduce Mike, I just hope that what we've done today is not to frighten you about a Frankenstein world that confronts us of something we can't control, but I hope quite the opposite, that we at Berkeley recognize the problem, and we at Berkeley have terrific people to address unique solutions to this global problem that's with us now and will become more increasingly with us on the horizon. So with that, Mike Botchin is our Dean of Biological Sciences, also Professor of Biochemistry. He's a distinguished member of our faculty, National Academy of Sciences member, American Academy of Arts and Sciences. His name is synonymous with understanding DNA replication. And with that, Mike, the floor is yours.
Well, thank you, Michael, and thank you, Art, Sarah, and Richmond, for sharing with us your work and a glimpse into how public health, molecular biology, and cell biology and chemistry will help us answer questions about drug resistance. As a dean, I'm always inspired by the excellence that we see in these events. It was just last week that I was talking about the existential threat of climate change and the need for sustainable materials and energy consumption. And this evening, at the risk of sounding like an alarmist, as Marletta just said, we don't want to be alarmists. We've been talking about drug resistance, a silent problem that the public doesn't think about often enough or deeply enough. But here's the thing. Hope lies in the fantastic minds thinking about and working on this problem, including the people our friends listening in have met here in this Zoom. We all need to be optimistic about the future, and I am, and understanding the great power of the experimental method and the culture of science. In the context of the future, I have to point out the incredible undergraduates and graduate students who study these problems and work side by side in the labs and in the field with our faculty scientists on these big questions and global challenges. And you've seen some pictures of them tonight. Lab pictures. I feel very strongly that while it's essential to apply our science to solve such problems, application and translation into practice goes hand in hand with foundational work that leads to those solutions. Basic science has always been one of our greatest strengths here at Berkeley, and it is a theme running through all of the events that we have in this series, for sure, through the talks we've heard tonight. I want to say and give a special thanks to you, our alumni and friends, for gathering on the Zoom. You join these fantastic minds by coming and listening, and to learn by becoming aware of the great powerhouse of science that we have here at Berkeley. Your support and advocacy for us is means everything to our community. And we're extremely gratified for your continued interest in us. If there's anything you want to learn more about, or if you'd like to support our work, for which there's a great need, please be in touch. We absolutely want you to be part of advancing basic science and discovery at Berkeley. So with that, fiat lux and go Bears.